Good afternoon, everyone, and welcome to our second session of the fourth day of Varsity Sci. Today we have three great cell biology talks lined up for you. The first talk, which is by Milena, will be live streamed, but because it will contain some results that have not been published yet, it will not be available online after this session, so definitely do join in and listen up because it won't be available. And this talk will be on the evolutionary origin of RNAs as regulators of DNA replication. Then we will have a talk from Zaki on the role of lipid signaling in the regulation of a particular type of channel. And after that, we will have a talk that is going to be pretty fly, so to speak, because it's on Drosophila and a member of the Drosophila centriole inventory. And that will be from Pallavi. Without any further ado, I will now pass on to...
Thank you once again, Milena, for your talk. And we will now move on to the second speaker, who is Zaki from Oxford. So he'll be talking about the role of lipid signaling in regulating type 1 cell task channel and mediating type 1 cell hypoxic inhibition. So without further ado, let's now welcome Zaki.
Hello. Hello, can you hear me?
Yeah, we can hear you.
Thanks so much for having me. I'm Zaki. I'm a DPhil student in the Department of Physiology, Anatomy, and Genetics at Oxford University. I have joined Oxford last year after completing my master's in the University of Alberta in Canada. My master's studies were in the area of respiratory physiology, and still here during my DPhil, my research is still within the area of respiratory physiology. Today, I will be talking about the carotid body and the peripheral chemoreception. So I'll start with a brief introduction of peripheral chemoreception, then I'll talk about the research question and objectives of my research. Then I'll talk briefly about the methods, then I will show a bit of examples about my preliminary results. So peripheral chemoreceptions are cells that are responsible for detecting the levels of oxygen, carbon dioxide, and blood pH, and then sending the signals to the cardiovascular and the respiratory centers in the brainstem. So their function is really important, and they play a very important role in controlling the respiratory parameters. There are two main peripheral respiratory chemoreceptors, one in the vicinity of the carotid artery, which is called the carotid bodies, and the other one is located in the region of the aortic arch, and so-called the aortic bodies. My talk today will be mainly about the carotid bodies, and here you could see they are located like just below the head, and they are bilateral and between the carotid artery bifurcation. But striking about these small organs is that they have a very high blood flow per kilograms. So it's 2 liters per minute per 100 grams, and they are very sensitive to changes in oxygen levels in the blood. So they will respond to oxygen levels that doesn't affect any other cell types. Carotid body is consists of two different types of cells. Type 1 cells are called Glomus cells. They are the main oxygen detector cells. And the other type of cells is type 2 cells. And type 2 cells actually are similar to the glial cells of the nervous system. So their role is mainly supportive to type 1 cells. Now I'll talk briefly about the characteristics of type 1 cells. So one of the main characteristics of type 1 cells is that they have a very large background potassium conductance under resting condition. And most of this background potassium current is carried out mainly by task 1 and task 3 channels. Also, type 1 cells express other types of ion channels, such as voltage-gated channels and calcium-activated channels. Task 1 and task 3 channels are members of the two-pore domain potassium channels. They have two pore-forming domain and four transmembrane domains and two extracellular cap helix, and they are responsible again for maintaining the resting cell potential of type 1 cell. So how type 1 cells respond to hypoxia? Actually, whenever there is reduction in the blood oxygen levels, the first thing that would happen is inhibition of background potassium current, and which means inhibition of task channels. So once the background potassium current is inhibited, the cell will depolarize. After type 1 cells depolarize, there is calcium influx followed by neurosecretion of different neurotransmitters, and the final outcome will be increased ventilation or tachycardia or induced systemic vasoconstriction. So what I want you guys to remember from this cascade is that whenever there is hypoxia, the first event is reduction of task channels, and the second event is calcium influx. So the question was, how type 1 cells detect hypoxia? There have been two main models proposed in order to answer that question. The first one is the membrane model. Here in the membrane model, the idea is that whenever there is hypoxia, it will affect the membrane conductance of type 1 cells, which will lead again to calcium influx and neurosecretion. Whereas in the metabolic model, this model actually is based on the fact that the mitochondria of the type 1 cells is quite different because they have a very low oxygen binding affinity. So the hypothesis, the idea here is that whenever there is hypoxia, there will be inhibition of mitochondrial function. And then once the mitochondrial function of type 1 cells is inhibited, there will be a reduced in the production of ATP and increased production of ROS and increase the production of lactate. Then all of these three events will lead to depolarization of type 1 cells. Another interesting thing was the capability of task channels to be regulated by G protein coupled receptors. The idea here is that once the G protein coupled receptor is activated, it will activate the phospholipase C, which in turn will cleave the BIP2 from the cell membrane into DAG and IB3. We don't really know what the consequences of this G protein coupled receptor activation on task channels. The idea is that it will inhibit task channels, but we don't know exactly what mechanism is that due. So it has been proposed it's either the activated G protein coupled receptor act directly on task channels, or whether it's the PLC activation or BIP2, DAG, or IB3. The other possible mechanism of GQ protein coupled receptor mediated inhibition of task channels is that endocytosis. So the idea here, once the G protein coupled receptor is activated, the task channels will translocate from the cell membrane to the cytoplasm. I will show you now a couple of data on both pathways. Here you could see in this graph, we could see that current of task 3 channels, and here they applied U73122, which is PLC inhibitor. And you could see once they inhibit phospholipase C, oxytremorin, which is a muscarinic agonist, which acts through inhibition, which acts through stimulation of G protein coupled receptor, is no longer able to inhibit task channels. Because if you compare between the blue line and the black line, the blue line is with PLC inhibitor plus the oxytremorin, and the black line is the U73343, which is inactive compound. So it's not supposed to inhibit phospholipase C. And again, when you inhibit phospholipase C and apply muscarinic agonist, task 3 channels are no longer inhibited. The other thing here, they tried to see what's the effect of DAG, and as I showed you in the previous slides, that DAG is one of the consequences of BIP2 hydrolysis, which is induced by phospholipase C activity. So here they tried to test what's the effect of DAG analog. So here they used the DIC8, which is DAG analog. So it's literally, it should be the same function and the same response on the cells. So once they applied the DIC8 with two different concentrations, they inhibit task3 channels in a similar way to the inhibition that's induced by muscarinic agonists. So here, we don't really know whether it's direct effect of DAG or it's secondary to activity of phospholipase C. So we show that DAG is able to inhibit task channels, but we don't really know whether it acts directly on task channels or not. This figure is on the effect of G protein coupled receptors on task channels, but here they looked at the internalization of task channels. And you could see here after a very short exposure to mascarine, both task, in particular task1 channels, translocate from the cell membrane to the cytoplasm after a very short application of mascarine. So it might be all of these three events together, or maybe task1 is more sensitive to endocytosis, whereas task3 is more sensitive to lipid signaling pathways. We don't really know. What's interesting is none of this work has been done on type 1 cells task channels, because these task channels were expressed in other cell lines. So what we wanted to do is to examine the effect of lipid signaling pathways on type 1 cells task channels. After that, we wanted to know what's the role of lipid signaling pathways in mediating hypoxic inhibition of type 1 cells task channels. So in order to do that, I started with doing calcium measurements experiments, because whenever there is an inhibition of task channels, the next direct step is elevation of intracellular calcium levels. So I wanted to start with calcium measurements experiments in order to get a general idea of what I would expect with patch clamping technique, and when I look closer into the cellular effect of lipid signaling. So what I usually do is that I dissect the carotid bifurcation from neonatal rats under terminal anesthesia. And here you could see that in the top left of this slide, this is the carotid bifurcation, then the glossopharyngeal nerve on top. So usually I dissect the whole thing and then decapitate the rat. Then I move on to another microscope where I do the micro dissection. And here you could see this is the carotid artery bifurcation, and the two branches of the carotid artery, and the small little artery in the back is the occipital artery. And you could see the carotid body is like a small collection, circular shape of cells. So once I dissect the carotid body, I start the enzymatic digestion in order to isolate type 1 cells. So I apply enzymatic solutions, and I place the cells in the incubator. After that, the cells were plated out on cover slips, and then for 2 hours. Then after that, I loaded the cells with into one dye, which is a fluorescent dye. Then I moved the cover slips into the perfusion bath. So the perfusion bath that I'm using now has been designed to allow only one solution to be delivered to the cover slips. So you could see in the top that the tube right there is the income. So it's the source of the flow. Then it goes to the superfusion dish, then out to the waste outflow. So this superfusate switch, actually it controls what solution will be delivered to the cells at this time. So only the solution in the middle will be delivered to the cells. So by changing mechanically the switch from the right to the left, I can control what solution I need to deliver to the cells, at this particular time. So usually solution A is my control solution, and solution B is the solution with any agent of interest that I want to examine type 1 cells response to. So now I'll show you a bit of my preliminary results. So here, this blue line is the intracellular levels of type 1 cells. So the idea is that we need first to test for hypoxia and see whether the cell will respond to hypoxia or not. In case the cell responds to hypoxia, that means this is for sure type 1 cells. So usually I start with testing for hypoxia. And here you can see once I test for hypoxia, there is an increase in the intracellular calcium levels. After that, I applied here, for example, a couple of potassium channel blockers. And it's DMP, LMP, or a cocktail of both. And you could see again with both DMP 543 or LMP 345, both will increase the intracellular calcium levels of type 1 cells. And also when I use the cocktail of both agents, there is even further increase of intracellular levels of type 1 cells. And again, here the idea is that when you block the potassium channels, you mimic the effect of hypoxia, so that will lead to calcium influx, and that's why we see here the increase in intracellular calcium levels. After that, I tried a couple of muscarinic agonists, and again here I tested first for hypoxia, then I applied oxytremorin, then muscarine chloride, then methacholine chloride, and with both I got increase in intracellular calcium levels. So what I'll be doing next week is that I will start again with muscarinic agonists, but this time I'll apply phospholipase C inhibitors and see whether I will get the same response with muscarine, or it will be inhibited when I co-apply muscarine with the phospholipase inhibitor. That's what it's for my presentation. Thanks so much for listening, and I'm willing to answer any questions.
Thank you, Zaki, for your talk. So once again, if you have questions, you can ask in person on Zoom, or you can post them, send them to me. Okay, there's one question on Zoom asking, saying that you mentioned that there are some hormones and neurotransmitters that can stimulate the GQ-PCR channels, causing the TAS1 channels to be inhibited. Could you give an example of those neurotransmitters and hormones?
For example, muscarinic agonists, they act through G protein coupled receptors, and they inhibit TASC channels.
Thank you.
Thank you.
So there's one question asking, are there any other potential functions? I think perhaps the person would like to clarify. Perhaps it's the functions of the TASC1 channel, if I'm not mistaken.
Mainly TASC channels, either in type 1 cells or in other cells, they are responsible for maintaining the resting potentials. So the idea of cell depolarization is that once you inhibit the resting potential, then that will lead to depolarization of the cell. So that's why here with type 1 cells in the carotid body, we see that during hypoxia, there is an inhibition of the background potassium channels. So once the resting potential is inhibited, then the cell will have the potential to depolarize. So their main function actually is to maintain the resting potential of different types of cells.
Thank you.
Thank you.
And there's one question from Zoom. Do you think that there could be different methods of regulating the response depending on the time scale? For example, via gene expression regulation?
In terms of type 1 cells, you mean? Or? I think this question.
Or should I repeat the question?
Yeah, please.
Do you think that there could be different methods of regulating the response depending on the time scale? For example, via gene expression regulation? Okay, so the person clarified in terms of TASC channel regulation.
I'm not quite sure that I really understand the question, but if it's asking about like if we play with different genes, because there have been a couple of experiments where people try to knock out either TASC channel genes and try to see the hypoxic responses. But the problem with that is that regulation of hypoxia and the hypoxic bodies is really essential. So even in knockout experiments, the body will compensate in that some way or another.
Thank you. Oh yes, so the person said you answered the question. Thank you.
Thanks.
So I think if there are no more questions, we will move on to the second talk. Thank you once again, Zaki.
Thank you. Thank you so much.
Oh, sorry, the third talk, sorry. So the third talk will be presented by Pallavi. She will be telling us about a forgotten member of the Drosophila centrole inventory. And so without further ado, let's welcome Pallavi.
Okay, just give me a moment. Is part of the slide obstructed by the Zoom window, or you don't, I mean, you guys don't see it?
No, everything's fine.
Everything's fine. Okay, so very good afternoon to everyone. I am Pallavi, a PhD student at the Department of Genetics here in Cambridge. Before I start, I'd like to say a very big thank you to the organizers for the Oxbridge Varsity Science Symposium. It's a wonderful initiative bridging science and research between Oxford and Cambridge, especially among the students starting right from the undergraduate level. So today my talk is going to actually introduce to all of you about centrioles and centrosomes in Drosophila. And I'm specifically going to speak about a centriole component known as RCD4 based on findings that we have from a very recent publication about characterizing RCD4's role in the Drosophila centriolar context. So starting off with a very brief introduction into centrosomes. Theodore Boveri is known to be the father of centrosome biology, but the first drawings of centrosomes actually date back to 1876 and 1883 from Van Boveri in some of his publications. But back then, this was still during a time when centrosome was not discovered yet. It was not until 1887 when actually two hallmark papers were published by Theodore Boveri as well as Van Benedin, which for the first time actually highlighted the presence of these spindle pole associated structures. So this image here that I'm indicating with the mouse cursor is actually one of the first few images from these publications where the main focus was actually about the early divisions that take place in the embryos of the parasitic nematode. So it was during these particular studies that both of these scientists actually for the first time observed and highlighted the presence of these structures at the spindle pole ends. So even though Van Benedin was one of the first few to actually observe and draw it out in his publications, it was Boveri who coined the term centrosome that has lasted us all the way up till now. Then in addition, it was not until several years later, about 12 years later, that that Boveri actually identified smaller granule-like substances within these centrosomes that are indicated here in the diagram or in the images here, which he then later on termed them as centrioles. So this is how the term centriole and centrosomes actually came about. So centrioles are dual functional organelles that organize centrosomes in dividing cells and also transition into basal bodies that template the formation of cilia and flagella. So a pair of centrioles, one of which is a mother centriole and the other a daughter centriole. So both of these centrioles differ in terms of their age and maturity, where the mother centriole is older and the daughter centriole is much younger, that is just developing at that point of time. So this pair of centrioles are surrounded by a matrix of proteins that is known as the pericentriolar material, otherwise known as the PCM as indicated here. So together, these pair of centrioles and the pericentriolar material make up the centrosome. And the centrosome functions as the major microtubule organizing center in dividing cells, where it plays an important role in the assembly of the mitotic spindle during cell division. But in cells that are not dividing, what happens to these centrioles is that they migrate to the cortex of the cell, where they transition into basal bodies and they template the formation of the primary cilium, or in certain cases, different types of cilia and flagella. The Drosophila is an excellent model system to study centrosome as well as cilia biology. So many of the studies that focus on centrosomes are actually conducted in the early Drosophila embryos, where very rapid mitotic divisions take place that are supported by centrosomes, as well as in the larval wing imaginal discs, as well as in the larval neuroblast. So the reason why Drosophila is such an attractive model to study centrosomes and cilia is primarily because due to the diversity of centrioles that are present in the various tissue types found in Drosophila, as well as in the different developmental stages of the fly. Whereas if we're interested in studying basal bodies or ciliogenesis, then we generally look at the type 1 ciliated neurosensory cells, for instance, these here, or specific cell types in the male germline, particularly the primary spermatocytes or the spermatids. The cardonal organs are actually an example of these type 1 ciliated neurosensory cells, which serve as an excellent model to study ciliogenesis. And cardonal organs can be found, for instance, in the Drosophila leg, as well as in the fruit fly's antenna. So central duplication is very closely tied in with the cell cycle. And centrioles only duplicate, and very strictly only duplicate, once per cell cycle. So central duplication begins at the end of the mitotic phase in late telophase. So as indicated by these black arrows here. So this is these green dots are basically the seeds or the initiation of the new centriole that's developing. Otherwise, it's also known as the procentriole. So throughout most of interphase, these new developing centrioles or the procentrioles continue to elongate, such that towards the end of interphase, they actually reach a size that is similar to that of the mother centriole. So initially, central duplication begins with the existence of a mother centriole, the older mother centriole, and the new centriole here that forms is termed a procentriole. So it's termed a procentriole all the way until just before mitotic entry. So once the centrioles have fully elongated and just before it enters mitosis, that's when we call it the daughter centriole. Many of the important stages or steps in central duplication actually take place during the cell's passage through mitosis. And one of the most important steps is actually centriole to centrosome conversion. So at the onset of mitosis, this daughter centriole recruits certain molecular components that actually convert it into a centrosome, and this centriole to centrosome conversion of the new daughter centriole enables it to recruit its own pericentriolar material that's marked by this red matrix here, and then for it to be able to duplicate itself in the next cycle. Simultaneously, during mitosis, the centrosome also undergoes centrosome maturation, and centrosome maturation is defined by the extensive expansion of the pericentriolar material surrounding the centrosome, and this is what supports its ability to nucleate microtubules and support the formation of mitotic spindle. So the pericentriolar material is defined by, again, this matrix of proteins, red proteins that surround the centrosome. So these images on the left here are represented by actual super resolution microscopy images of centrioles from cultured Drosophila cells. So as I explained, this centriole here is the mother centriole, and this dot, the green dot at the side, is actually the developing procentriole. So in interphase, we have the procentriole here, and as the cell progresses through the cell cycle, we can see that this daughter centriole now is slowly transitioning into a centrosome as it undergoes central to centrosome conversion, such that at the end of the M phase and late telophase, that daughter centriole is now able to duplicate to initiate the next cycle of centriole duplication. So, so over the years, concerted studies in several model organisms, including the C. elegans, Drosophila, as well as in human cultured cells, have identified a set of core proteins or a framework of proteins that are actually involved in centriole duplication. And PLK4, known as polo-like kinase 4, is believed to be the master regulator of centriole duplication that initiates the entire cascade of steps that are involved in centriole duplication. So, we now do have an idea of the most important components that are involved in centriole duplication. But the question remains that could there actually be additional components that are involved in centriole duplication? So, in response to this question, there were two genome-wide RNAi screens that were conducted in Drosophila, namely by Goshima and authors and Dobbelaere authors about 10 to 12 years ago. And this identified, or these screens identified, several new components that were yet to be characterized. So at this point of time, 10 years down the road, several of these components have been characterized. For instance, ANA1 here is now known, for example, ANA1 here is now known to play an important role, whereas there's still some other components that are not well studied or that are still unexplored. And of these several components, RCD4 is one such component that remained unexplored, which is why my project focused specifically on RCD4 and identifying the role that it plays in centriole duplication or centriole biogenesis. So as RCD4 was previously unstudied or unexplored, I began my project by generating CRISPR-Cas9 mediated, or I conducted CRISPR-Cas9 mediated mutagenesis to generate RCD4 mutant flies. So my mutagenesis resulted in two deletion alleles, one that's termed as RCD41 here, that deletes about quite a big chunk of RCD4, but importantly, the start codon is it's still present in the remaining deletion, and about 1/3 of the C-terminal end of the gene is still present. And then the second one was an RCD42 allele, which more or less eliminated the start codon as well as the vast majority of the gene. So these are just some phenotypic, these videos just represent some phenotypic observations of how these flies look like. So as you can see, well, the flies, at least in RCD42, are still alive. If you carefully see, they're able to move their legs, but it's very minimum. Whereas in RCD41, even though the flies are very slow and uncoordinated, they're still able to mildly move around or walk around. So this indicated to us that the RCD41 allele was very likely a hypomorph. It functions as a hypomorph. And this remaining residual portion here that is not deleted from the gene potentially has some function, which is why we see the difference in severity between the phenotypes of these two mutants. So one of the phenotypes that we observed was that embryos derived from the mutant females of RCD4 were actually sterile. So as you can see here, none of these mutant females, either from RCD41 or RCD42, were able to give rise to any progenies. So let me give you a little bit of a background in terms of embryos and their connection to centrioles and centrosomes. So embryos, the first 13 to 14 rounds of mitotic division in the Drosophila embryo occurs in syncytium, where there's no cell boundary between the different nuclei, so they all share a common cytoplasmic pool, as you can see here in this image. So there's no cell boundary all the way up till cycle 13, and the mitotic divisions are extremely rapid, alternating between the S phase and the M phase. So in order to keep up with this very rapid rapid mitotic divisions and for the faithful segregation of the nuclear content, centrosomes play a very important role in the early divisions of Drosophila, such that they are able to support the formation of the mitotic spindle. So in the absence of centrosomes, this is when mitotic failure takes place early on in the Drosophila embryo, which then affects the development of the embryo. So to understand why the embryos derived from RCD4 mutant females were sterile, I collected some of these embryos and I analyzed them via immunofluorescence. So as you can see from the images here, in the wild type, they're very nicely organized mitotic spindles with centrosomes that are present at the spindle poles of these mitotic spindles. Whereas when we look at RCD4-1 and RCD4-2, it's very obvious that there is drastic mitotic defects where the spindles have a very odd or very globular shape. There are free microtubules that are observed. And when it comes to RCD4-2, which is the null mutant, mitotic divisions don't really proceed very long, or don't proceed further. But most importantly, it's important to realize that actually none of these spindles actually show the presence of any centrosomes. So the spindle poles lack the presence of any centrosomes at all. So this explains that due to the centrosome loss that's taking place in the embryos, this leads to or gives rise to the mitotic defects that we observe. Then going back to the video that I had showed earlier, you would have noticed that the flies are very uncoordinated. They were not really able to walk or fly for that matter. So coordination in flies or locomotor coordination in flies is partly controlled by the cardonal organs, as I had mentioned earlier. And these cardonal organs are actually ciliated neurosensory cells, and they are found in the antenna as well as in the Drosophila leg. So this is the fundamental unit of the cardonal organ, which is made up of two neurosensory cells, where this is the cell body and the basal bodies. So as I'd mentioned earlier, centrioles, when they're not dividing, when the cell is not dividing, the centrioles actually migrate to the cell cortex, where they then transition into basal bodies and function in celiogenesis. So in these terminally differentiated neurosensory cells, the basal bodies are indicated here in red, and that's how they template the formation of cilia that you see in orange here. And these basal bodies are connected to the cell body via a structure known as the ciliary rootlet. So any defects that would be, any centriolar defects would have a consequence on the basal bodies, and as a result affect celiogenesis and the function of these cardonal organs. So given that the flies or the mutants that I have looked uncoordinated, I did a coordination test to actually see how far were the flies able to climb in a given duration of 30 seconds. So in comparison to wild type and the rescue flies, both RCD4-1 and RCD4, their mobility or their climbing ability is severely compromised, where the phenotype is much more stronger in RCD4-2, which is the null mutant. So again, to get a better insight into what exactly is happening at the structural or the basal body level in the Drosophila leg, that's when I dissected and visualized these cardonal organs that I had mentioned earlier. So marked in red here are the basal bodies, and as I'd also discussed earlier, here in green are the ciliary rootlets that link the basal bodies to the cell body. So in wild type, we see a very nice arrangement of the cardonal organs or these neurosensory cells, with each unit having two basal bodies. Whereas when we look at what's happening in RCD4 mutant flies, though there are still some, there's still some signal in red, which indicates that there are some basal bodies, but it's highly disorganized. And instead of seeing two distinct signals representing two basal bodies, here we see a diffuse signal. And the phenotype is then further aggravated in the null mutants in RCD42, where again the cells are severely disorganized, and here the signal doesn't even seem to be very prominent, indicating that there's potentially the absence of any basal bodies in these mutant flies. So, well, these are immunofluorescence images, and they give us an indication based on antibodies that target the centrioles. But to get an even better understanding, that's when I actually moved to doing electron microscopy. So these images here are transverse sections along these transverse sections that cut through podotinal organs at this particular slice. So in each of these, as shown here, these two structures here represent the cilia, or rather the exonym that protrudes out of the ciliated cells. So in wild type, corresponding to what we see in the immunofluorescence images, there are actually two cilia that we observe, whereas in RCD41, the vast majority of the cells either just had one or they had none. So that indicates here why we don't actually see two clear signals corresponding to the basal bodies. And then when we move on to RCD42, the electron microscopy result very clearly explains why we don't see any signal. That's because there are basically no basal bodies that template the formation of any cilia. So these results indicate that the coordination defects that we observe in the RCD4 mutant flies can be attributed due to the progressive loss of centrioles and consequently basal bodies in these ciliated neurosensory cells of the podotinal organs. So, so at this point of time, now we have a brief understanding of what are some of the phenotypes of RCD4 mutants, or what are some of the phenotypes that we see in the absence of RCD4 in flies. The next question that we had was then, given that RCD4 is an unexplored centriolar component, we were next interested to see how RCD4 might interact with other centriolar components. So for this, I conducted an affinity purification of components from cultured Drosophila cells, and then did mass spectrometry to identify which other centriolar component might be associated with RCD4. And our mass spectrometry results indicated that ANA3, so ANA3 is another known centriolar component. So the results indicated that ANA3 was the only other centriolar component that appeared to be pulled down along with RCD4 in these affinity purification experiments. So ANA3 is actually a very large protein that spans about 200 amino acids, and it's about 250 kilodalton in size. So now that the mass spec results did indicate that RCD4 and ANA3 pulled down together, this then implied or suggested that maybe both of the proteins might actually be interacting together. So to address this question, and given that ANA3 is a very large protein, what I did was I created truncations of the ANA3 into different fragments so that it's easier for us to see which particular fragment, and hence to narrow down the region or the binding region between RCD4 and ANA3. So in relation to this, we conducted some in vitro binding assays where I mixed fragments of, or where I mixed the RCD4 protein with the different fragments of ANA3 and tried to identify which of this combination resulted in a complex that And as seen from these gel images here, and as well as Western blot, it's very evident that this particular fragment here, C1, is the fragment that RCD4 interacts with and forms a complex. So this brought us to the conclusion that there is a direct interaction. So it confirmed our mass spec results and showed that there's a direct interaction between RCD4 and specifically the C-terminal half of ANA3, and that these two proteins interact to form a subcomplex. So now that we know a little bit more about what the phenotypes of RCD4 mutants are, what is the binding partner of RCD4 when it comes to the centriolar components, the next question was then to visualize where exactly in the centriole does RCD4 and its binding partner NR3 fit in. So before I discuss those results, I'd like to very briefly introduce you to the centrosome architecture. So as I mentioned earlier, centrioles are surrounded by the pericentriolar material, which together makes up the centrosome. So here we have categorized the centrosome into different zones, where zone 1 and zone 2 make up the centriole, whereas zone 3 and zone 4 make up the pericentriolar material. So within zone 1 and 2, zone 1 is defined to be the core of the centriole, the very middle part or the very inside part of the centriole, whereas proteins that localize to zone 2 are more on the periphery of the centriole. And even though zone 3 and zone 4 are both known as the pericentriolar material, proteins that surround the centriole, the proteins that localize to zone 3 are known to be, they function as a bridge or an interface between the centriole as well as the pericentriolar material components that are found in zone 4. Additionally, there's an extra zone known as zone 5 here, and this usually sits at the distal end or at the top of the centriole, and it functions as a cap or a plug at the very end of the centriole. So a centrosome is made up of the mother centriole, as you can see here, and also made up of the daughter centriole. As the daughter centriole is still developing, it only consists of zones 1, 2, and 5, but it lacks zones 3 and 4, which are essentially the pericentriolar material. And I will go into the details of, so it's during centriole to centrosome conversion, as I mentioned earlier, that is exactly when the daughter centriole converts into a centrosome for it to be able to then recruit proteins that make up the PCM, which are essentially zone 3 and zone 4. So moving into the results, so I looked at centrioles, or rather I conducted super resolution microscopy of centrioles from cultured Drosophila cells and tried to identify where RCD4 and NR3 localize within this zonal architecture of the centrosomes. So I stained RCD4 and NR3 in respect to different markers that are responsible for the different zones. So here, for instance, SAS6 here is a zone 1 protein, which is why it's present in the middle here, and also on the daughter centriole at its side. SPUD2 here is a zone 2 marker, Astellas zone 3 marker, and CP110 a zone 5 marker, which you can see is at the very end, the distal end of the centriole. So these results very clearly indicated that both RCD4 and NR3 localize to the core of the centriole in zone 1. And as both of the proteins bind and interact together and are believed to form a subcomplex, it makes sense that both their localizations are similar when it comes to the zones of the centrosome. So as I was mentioning earlier, centriole to centrosome conversion. So centriole to centrosome conversion is the process that the daughter centriole undergoes before it is able to be licensed to be able to organize its own pericentriolar material and for it to be able to duplicate in the next cycle. And this particular process is driven by the recruitment of a molecular network of centriolar components known as CEP135, ANA1, and Astellas. So these three proteins interact end to end to form a molecular network. And once these three components are recruited to the centriole, that is what gives or licenses the daughter centriole for it to then proceed on with its own duplication in the next cycle. So what our results indicate when it comes to, or rather when we knocked down RCD4 and NR3 in cultured Drosophila cells, what we realized that the knockdown of both of these proteins actually affected the recruitment of ANA1. And as shown here, ANA1 is one of the components that is involved in this molecular network that plays a role in central to centrosome conversion. So these results that indicated that in the absence of RCD4 and ANA3, in the vast majority of the cells, ANA1 was not being recruited to the daughter centriole, suggested to us that this subcomplex of RCD4 and ANA3 plays an important role or functions as a prerequisite in order for central to centrosome conversion to take place. So this brings us to the conclusions, indicating that RCD4 and ANA3, they interact to form a subcomplex. We have seen some of the phenotypic consequences of RCD4 mutant flies, and it shows that in the absence of RCD4, there's central loss, as well as it affects celiogenesis and basal bodies in the ciliated neurosensory cells. And in addition, in terms of the molecular mechanism, we see that both ANA3 and RCD4 here interact together, and they function as a prerequisite that is required for the recruitment of ANA1 onto the growing or the developing daughter centrioles for it to then subsequently undergo central to centrosome conversion. So that summarizes my talk, and I'm open to any questions.
Thank you, Pallavi, for introducing us to a very important component in central biogenesis in Drosophila. So I'm once again gathering questions from YouTube and Zoom.
Yeah.
So one question from Zoom is that, have homologs of RCD4 been found in other organisms such as humans?
I'll just move to the slide. So yes, so the homologue of RCD4 is actually called PPP1R35. It's a protein phosphatase 1's regulatory subunit called R35. And then the human homologue of ANA3 is actually called rotatin. And interestingly, both of these proteins, or rather specifically the homologue of ANA3 known as rotatin, is involved in microcephaly. So there are many clinical cases that are associated with mutations in rotatin. That's the human homologue of ANA3.
Thank you. If anyone has any questions, feel free to ask them in person as well. So perhaps one question from myself. So I'm just a bit curious because you mentioned that flies who don't have RCD4 show phenotypes such as uncoordinated movements. So because NR3 is similar to RCD4, do flies without NR3 also show a similar phenotype?
Yeah, the phenotypes are same in the embryos as well as in the quadrilateral organs. I mean, because they're partner proteins, it's quite similar. But what's interesting is that the phenotypes of NR3 are actually a little bit more severe than that of RCD4.
I see. Thank you.
Okay.
One more question. Does that C end-to-end protein network explicitly act as a scaffold for building the centrosome around it?
No, it doesn't. No, it's not really involved in, it's not really, that molecular network per se is not involved in building the scaffold of the centrosome, but definitely the fact that these particular centriolar components are recruited to the centriole, then it leads on to a cascade of other protein interactions and recruitment of proteins that then establish the formation of the pericentriolar material. But no, directly these three proteins are not particularly involved. But yes, indirectly it does lay the foundation that subsequently allows for the formation of the pericentriolar material.
Thank you once again.
Okay, thank you very much.
So if there are no more questions, I will pass the floor to Christoph to conclude the session. Thank you.
So thank you very much once again, Pallavi, Zaki and Milena, for 3 brilliant talks that did not only cover a wide range of topics from cell biology and from physiology as well as bits of biochemistry thrown in, but were very interesting in that they demonstrated a wide range of experimental methods and techniques that will surely be very interesting to anyone who is watching this talk right now or who might be watching this talk in the future to find out how these methods could be applied in their own work, how these methods could be applied for their PhD if they're undergraduates who are preparing to enter a PhD program. So I'm sure that data will be very informative to people who are looking to make those decisions about further study. So thank you once again to all of our wonderful speakers. What we're going to have after this talk at 4 p.m. is our keynote from Professor Jane Clark, who is the president of Waltham College, Cambridge, not Oxford, as well as, well, she's also the professor of molecular biophysics and an advocate for women in science. So I hope you'll all be able to join us later on at 4 p.m. And for now, it's goodbye from me. Thank you very much, everyone, and thank you for agreeing to speak with us.